Welcome again friends. So in this video lecture we are going to talk about restriction site. In our previous video we already have talked about restriction endonuclease. We have given a basic introduction of restriction endonuclease. What was the first restriction endonuclease purified, and we also talked about how we can nomenclature, or you can say name, a particular restriction endonuclease, or you can say how they are named. So in that video I have told you that restriction endonuclease are very specific in their action. They only cut a double stranded segment that have a specific nucleotide sequences of near about 4 to 6 base pair in length, and we have talked these specific sequences as restriction site or recognition site. So in today's video, and I hope two more videos, we are going to talk about this specific nucleotide sequences called restriction site or recognition site. Okay, so let's start. So in our very first video I have told you that we use restriction endonuclease to cut a target DNA sequence from a very large DNA, you can say heterogeneous DNA source, you can say. Okay, suppose we need to cut the gene that have the ability to code protein insulin. Okay, so we can we can apply restriction endonuclease that have the ability to recognize the specific nucleotide sequences of the insulin coding gene, and it will cut it. So that what restriction endonuclease or restriction enzyme is. Okay, so these restriction site are simply palindromic in nature. So what this palindromic sequence thing is, I am going to talk with you in this video. So let's start it. So restriction enzyme have the ability to make two type of cuts. One is blunt or staggered cut. Okay, so let's suppose this is your target DNA sequence from a large DNA molecule, and restriction enzyme will recognize this larger, that is target DNA sequence, and then it have the ability to make two kinds of cut. One is your blunt cut, another one is your staggered cut. Okay, any one, not both. Okay, so on the basis of this, after cutting the remaining target sequences out of the large DNA sequences are termed as restriction fragment, and on the basis of type of cut that was given by restriction enzyme, there are two ends. One is blunt end, another one is overhanging end. So what those are, we are going to talk about. Okay, so what we have learned, that restriction endonuclease are a nucleus that simply evolved by bacteria to prevent viral action. Okay, they have the ability to prevent entry of foreign DNA into the host cell. Okay, so this restriction endonuclease will have the ability to cut a double stranded DNA segment, you can say, which have specific nucleotide sequences, and this specific nucleotide sequence containing double stranded segment of which they have the ability to cut, we are going to talk here, and this is called restriction site, right? So in our previous video I have already discussed with you how we name this restriction enzymes. So let's repeat it once again, that the first word simply tell us the genus from which it is isolated. Second and third word simply tell us about the species. The fourth thing will tell us about the strain, and the number, that is usually in Roman, will simply tell us about the order of discovery of enzyme from that particular bacterium. So eco R5 is the number 5 enzyme that was isolated from that particular bacterium. Here the bacterium is Escherichia coli, right? So suppose the specific site, or you can say recognition site for Escherichia coli is this. Okay. So if the cleavage site is exactly on the axis of symmetry like this, then the resulting restriction fragment will have blunted, and the cut is called blunt cut. That means if if I am told, I am going to explain this in a very simple way. So simply they wants to tell us if the restriction endonucleus will cut this specific restriction site into equal part, then we will have blunted. Simple. But the technical term of that thing is the cleavage point occur exactly on the axis of symmetry. Okay, that means we will cut our restriction site symmetrically. That what they want to tell us. Okay, and the blunt cut simply generates blunted in the restriction fragment. So these things are restriction blunt in, and this type of cut are called blunt cut. Again, so I hope you have already figure out what overhanging hint are. Overhanging hint simply cut your restriction side asymmetrically in normal words. Okay. So the resulting fragment will have sticky end or cohesive end. So blunt cut have the ability to divide the restriction site into equal portion, or you can say to symmetrical portion, and those restriction fragment have blunt end, and in case of overhanging end the restriction site will be divided asymmetrically and they have staggered in, or you can say overhanging. Okay. Now this staggered end or overhanging end is of two types. One is 5 prime overhang, another one is 3 prime overhang. Now if the cleavage side is near the 5 prime end, then you will have 5 prime overhang, or you can say staggered end. Again, if the your cleavage side is near the 5 prime end here also, then you will have your 5 prime overhang or staggered end. If the cleavage side is near the 3 prime end, as I have mentioned here, then you will have 3 prime staggered end. So on the basis of cut made on the restriction site by restriction endonucleus, you have total 3 type of restriction fragment. That is what the remaining fragment, or you can say the component after treatment by restriction endonucleus on what restriction site. This restriction fragment are of 3 types. One is your blunting containing restriction fragment, and overhang end containing restriction fragment are of 2. One is 5 prime overhang and for 5 prime overhang end containing restriction fragment, another one is 3 prime overhang end restriction fragment. Okay, that was the main topic for today's lecture, and there is an additional thing too, that is star activity. Suppose in non-standard condition, that means if there is low ionic strength or high pH condition, some restriction endonuclease have the ability to recognize similar restriction site but not identical of their defined restriction site. It may sound like something cross reactivity that you have learned in immunology, right, where antibody have the ability to recognize identical similar epitopes but not identical epitopes of which they are specific. Okay, they and the similar thing is exactly in case of RDT is star activity, in which your restriction endonuclease have the ability to recognize similar restriction site not identical under non-standard condition, and this kind of altered specificity has been termed as star activity. In immunology, the same funda is going on between antibody and antigen, and we call them as cross reactivity. And in case of RDT, that funda is going on between our restriction endonuclease and restriction site under non-standard condition, that means low ionic condition and high pH condition, and we call them as star activity. This altered specificity has been termed as star activity. So I hope the today's lecture is clear to you, and if it is, please like this video, share with your friends and subscribe my channel. And if you need a lecture on cross reactivity, what the cross reactivity is, then please comment on the comment box. Okay, thank you. Bye bye.